I'm going to explain mitosis and meiosis. Mitosis is a phase of the cell cycle in which the spindle apparatus assembles, binds to the chromosomes, and moves the sister chromatids apart. It is divided into 5 stages. First, we start off with interphase, which prepares the cell for mitosis. Interphase has 3 parts to it: G1, S1, and 2. During G1, the cells undergo a major portion of growth. During the S phase, each chromosome replicates to produce 2 sister chromatids. In G2, the chromosomes coil even more tightly. You can see that the DNA has been replicated and the centrioles replicate, and the cell is ready to prepare for division. During prophase, the chromosomes condense and become visible. The chromosomes appear as 2 sister chromatids held together at the centromere. The cytoskeleton is disassembled and the spindle begins to form. Lastly, the nuclear envelope breaks down. The next phase is prometaphase. Here, the chromosomes attach to the microtubules. Then each chromosome is oriented so that the kinetochore of the sister chromatids are attached to the microtubules from the opposite poles. The chromosomes move to the equator of the cell. Next is metaphase. During this phase, all of the chromosomes are aligned at the equator of the cell. The chromosomes are attached to opposite poles and are under tension. After metaphase comes anaphase. Here, the proteins holding the centromeres of the sister chromatids are degraded, which frees individual chromosomes. The chromosomes are pulled to opposite poles and the spindle poles are moved apart. Then comes the last phase, telophase. Here, the chromosomes are clustered at opposite poles and they can decondense and the nuclear envelopes reform around the chromosomes. This one is the first round of cell division because homologous chromosomes separate and the daughter cells have only the haploid number of chromosomes. Meiosis 2 is the second round of division in which the 2 haploid cells undergo a mitosis-like division without DNA replication to produce 4 haploid daughter cells. Meiosis 1 is made up of prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, the chromosomes begin to condense and the spindle of microtubules begin to form. The DNA has been replicated and each chromosome consists of 2 sister chromatids attached at the centromere. Homologous chromosomes pair along the entire length during synapsis. Crossing over occurs which hold homologous chromosomes together. In metaphase 1, the pairs of homologous chromosomes align at the equator of the cell. Chiasmata keep homologous pairs together in microtubules from opposite poles the sister kinetochores of each homolog, producing tension. A kinetochore microtubule from one pole of the cell attaches to one homolog of a chromosome, while a kinetochore microtubule from the other cell pole attaches to the other homolog of a pair. In anaphase 1, kinetochore microtubules shorten and homologous pairs are pulled apart. One duplicated homolog goes to one pole of the cell and the other duplicated homolog goes to the other pole. Sister chromatids do not separate. This is in contrast to mitosis, in which duplicated homologs line up individually on the metaphase plate and sister chromatids are pulled apart in anaphase. In telophase 1, the separated homologs form a cluster at each pole of the cell and the nuclear envelope reforms around each daughter cell nucleus. Cytokinesis may occur. The resulting 2 cells have half the number of chromosomes of the original cell. Meiosis 2 is made up of prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, a new spindle apparatus forms in each cell, and the nuclear envelope breaks down. In metaphase 2, chromosomes consist of sister chromatids that are joined at the centromere and are aligned along the metaphase plate in each cell. Kinetochore microtubules from opposite poles attach to kinetochores of the sister chromatid, as in mitosis. Phase 2, when the microtubules shorten, the sister chromatids are pulled to opposite poles of the cells, just like mitosis. In telophase 2, the nuclear membranes reform different clusters of chromosomes. After cytokinesis, 4 haploid cells are the result. No two cells are alike due to the random alignment of homologous pairs at metaphase 1 and crossing over during prophase 1.